Hey everyone, ready to dive into something pretty amazing.
Absolutely.
Today we're taking a look at this groundbreaking research paper about AlphaFold 3.
AF3 for short.
Right, AF3. And it's seriously shaking things up in biology. We're talking about predicting the 3D shapes of those tiny molecules. You know, the ones that make life happen.
Like proteins.
Exactly. Proteins, DNA, even those tricky little ligands.
It's incredible. What gets me is that we're not just talking about single molecules anymore. AF3 can actually predict those intricate structures that form when those molecules get together.
It's like understanding a whole molecular dance.
Right. Think of a protein attaching to DNA.
Or maybe like a drug perfectly fitting into its target.
Exactly.
Before we get ahead of ourselves, let's back up for a second. Why is knowing the structure of these biomolecular complexes so important? It sounds really cool, but what's the real-world impact?
Well, imagine trying to fix a clock, but you don't know how the gears fit together.
You'd be lost.
Exactly. You might fiddle around, but you wouldn't really understand how it works.
Makes sense.
Biomolecules are like those intricate clockworks. Knowing their precise structure, that's the key to understanding how they function.
So how does AF3 compare to its predecessor, AlphaFold 2? I mean, the paper mentions some major improvements, but they're a little technical.
Well, let's start with this generative diffusion approach.
Okay.
Imagine you have a blurry photograph. AF3 kind of removes the noise from a random structure, slowly refining it, like until this clear 3D model emerges.
So it's more accurate.
Way more accurate, and it's also more confident in its predictions than AF2.
Interesting. So it's like starting with a rough sketch, and then you carefully fill in the details until you have this masterpiece.
Exactly.
Now, I also read that they simplified the architecture of AF3. They got rid of something called an Evo former. Was that a risky move?
You know, it was a gamble, but wow, did it pay off.
Really?
Yeah, they realized that removing that complex component made AF3 much more efficient and adaptable. And the best part, no loss in accuracy.
Wow. So sometimes less is more.
Yep.
Now, the paper also mentioned that AF3 can predict the exact positions of individual atoms. That's different from AF2, right?
Right. AF2 worked with more abstract representations.
So why is predicting individual atom positions so important?
Well, think of it like this. It's like going from assembling a puzzle with big chunky pieces to working with these tiny, precise components.
So it's a much finer level of detail.
Exactly. And that finer detail allows AF3 to tackle some really, really complex structures.
Okay, so we've got a more accurate, efficient, and versatile tool. But the real test is how it performs, right?
Yeah.
Out in the field. I mean, the paper highlights some impressive examples of AF3 tackling these different types of biomolecular interactions. Where should we start?
Let's dive into protein-ligand interactions.
All right. Protein-ligand interactions.
Now, these are absolutely crucial for drug design, you see, because drugs often work by binding to specific proteins in our bodies.
Right.
Now, the traditional tools for predicting these interactions, they really struggled, especially with complex ligands.
Ligands, those are the ones with the...
The twists and turns.
Yeah, twists and turns, like a molecular pretzel.
Exactly. And AF3, well, it just blows those tools out of the water, even without knowing where the ligand is supposed to bind.
No kidding.
Yeah, they actually put it to the test. They did this benchmark called Posebusters, and AF3 aced it. It even predicted the position of this really complex ligand called 5S6S07-sulfo-DADH.
That's a mouthful.
Oh, it is. It's a molecule with tons of branches and rings, but AF3 handled it with ease.
Wow. So what about those interactions that involve DNA and RNA? You know, those genetic molecules, the blueprints of life.
Oh, AF3 excels in this area too. It even outperforms those specialized tools designed specifically for that task. And here's the kicker. It can handle both small and massive structures.
So it's not afraid of a challenge.
Not at all. The paper actually highlights AF3's prediction of the human 40S small ribosomal subunit, and that's massive, over 7,000 residues.
That's going from modeling a tiny padlock to mapping out like a giant skyscraper. Incredible. And can it handle those modifications too? You know, those tiny tags that can completely change how proteins behave.
Absolutely. Think of those covalent modifications like adding sticky notes to a protein. They can change how a protein functions or how it interacts with other molecules.
Right.
And AF3, it can predict where those modifications are with amazing accuracy.
So we get a much deeper understanding of how these little tweaks can have a huge impact.
Precisely.
Okay, so we've talked about AF3 mastering individual interactions. But how does it handle predicting those complex protein complexes, those molecular machines that drive so many cellular processes?
It's amazing at that too. AF3 surpasses its predecessor in predicting how proteins fit together, like puzzle pieces. And this improvement is especially important for understanding antibody-protein interactions.
Those are crucial for our immune response, right?
Exactly. Think of antibodies like guided missiles targeting those invaders. AF3 helps us predict exactly how they lock onto their targets.
This is incredible. We're talking about a tool that could revolutionize how we understand and manipulate life itself.
It really is.
And on top of all that, AF3 can assess its own confidence in its predictions. It's like having a built-in quality control system.
Exactly. It can tell you, hey, I'm pretty sure about this one, or take this one with a grain of salt.
That's incredibly valuable.
Mm.
Researchers can focus on the most reliable predictions, right? And guide their experiments much more effectively.
Absolutely.
But we need to be realistic, too. No model is perfect. And the paper does mention some limitations of AF3.
True.
So let's dive into those next. All right, so we talked about how amazing AF3 is, but like we said, it's not perfect.
Yeah.
The paper mentions these challenges with something called stereochemistry. What's that all about?
Well, stereochemistry really comes down to the way atoms are arranged in 3D space within a molecule. It's like building with Legos, you know?
I see. If you put the blocks together in the wrong way.
You won't get what you're trying to build.
Right.
And AF3 can sometimes have trouble with this thing called chirality.
Chirality. Okay.
It's like, oh, your hands, they're mirror images of each other, right?
Right.
And some molecules, they also have these mirror image forms.
Interesting.
But if you get the handedness wrong, it can totally change how the molecule works.
Oh, I see.
So sometimes AF3 might predict the structure with the wrong chirality.
Oh, wow.
Or even put atoms in places where they clash, you know, overlapping in ways that just aren't physically possible.
So it's like trying to force two puzzle pieces together that just don't fit.
Exactly.
How big of a problem is this for the researchers who are using AF3? Could it lead them down the wrong path?
It's definitely something to be aware of, but thankfully the researchers are working on it. They're using these techniques, like ranking penalties. It's like giving AF3 a little nudge.
Like, hey, pay attention to this.
Exactly.
Yeah.
Saying, hey, these structures over here, they're less likely to be right. Try to avoid them. Okay.
So AF3 is learning from its mistakes. Another limitation that caught my eye in the paper was something called hallucinations. Is AF3 seeing things?
Well, not exactly seeing, but sometimes it can get a little carried away with its predictions. You know, there are regions of a molecule that are actually kind of floppy, like a wiggly chain.
Flexible.
Right, flexible. And AF3 might predict this nice, well-defined structure where there shouldn't be one.
Oh, I see.
So those hallucinations, they can be a bit misleading.
Because
they can make a protein or complex look more rigid than it really is.
So it's like AF3 is embellishing the picture, adding in details that aren't really there.
Exactly, like a molecular artist taking some creative liberties.
So how do researchers know if AF3 is hallucinating?
Well, that's where those confidence measures come in.
Ah, okay.
AF3 is more likely to give a lower confidence score to the regions where it might be hallucinating.
It's like it's saying, I'm not so sure about this part.
Exactly. Don't take my word for it.
Don't take it as gospel. So it sounds like the research team is aware of these limitations.
Oh, yeah. They're definitely working on making AF3 even better.
And what are they doing to improve it?
Well, they're exploring some cool techniques like this distillation training.
What's that?
AF3 actually learns from its predecessor, AlphaFold 2.
Interesting.
Yeah, and AlphaFold 2 was a lot less prone to those hallucinations. It's kind of like AF3 is getting some advice from its older, wiser sibling.
Okay, let's shift gears a bit now and talk about some of the real-world applications of AF3. The paper is full of amazing examples, but one that really stood out to me was the story of Donson.
Ah, yes, Donson, a perfect example of how understanding the structure of these complexes can lead to some real breakthroughs.
Yeah, breakthroughs in how we understand things like biological processes.
Absolutely.
Now, for those who might not be familiar with Donson, can you give us a quick rundown?
Sure. So Donson, it's this protein, and it has a vital role in DNA replication. You know, that process where cells copy their genetic material.
Right, before they divide.
Exactly, and it's essential for life. But for the longest time, scientists were scratching their heads about how Donson actually fit into this whole complex process.
They knew it was important, but they didn't know what it actually did.
Right. So how did they solve the mystery?
Was AF3 involved?
Oh, absolutely. AF3 was key here. Researchers used it to model how Donson interacts with these other proteins that are also involved in DNA replication.
And what did they find?
Well, they found that Donson is involved in the very first step of DNA replication. It's called replication initiation.
Wow.
Yeah. Pretty cool, huh? It's like AF3 gave them this backstage pass to the molecular machinery of DNA replication.
That's a great analogy.
And this is just one example, right? AF3 is helping us unlock all these secrets of life.
Absolutely. Another area where AF3 is having a huge impact, I think, is in drug design. The paper mentions LGK974. That's a clinical stage inhibitor.
Oh, yeah, LGK974.
It targets a protein that's involved in something called the Wnt signaling pathway.
Right, the Wnt pathway. It's kind of like a cellular communication system. It's
Mhm.
involved in all sorts of things like, embryonic development and tissue regeneration.
But in some cancers, this pathway goes haywire.
Right, exactly. It's
Yeah.
like the communication system starts sending the wrong signals, causing cells to just grow out of control. And that's where LGK974 comes in.
So it blocks the signals.
Well, it's designed to block this key protein called PRCN. It's essential for that Wnt pathway to function properly.
Okay.
But here's the thing, PRCN doesn't act alone.
It has a partner.
It forms a complex with another molecule called the WNT3A peptide.
A 2-for-1 deal.
Exactly. And this is where AF3 really shines. Researchers used it to model the structure of this PRCN WNT3A complex in such detail.
So they can see exactly how they fit together.
Right. And because of that, they were able to design LGK974 to fit perfectly and block the active site of PRCN, basically shutting down that Wnt pathway in cancer cells.
So it's like designing a key that fits a specific lock, and only that lock. That's incredible. And by targeting the complex so precisely, I'm guessing that it reduces the chances of the drug interfering with other important cellular processes?
Exactly. Less side effects. It's a great example of how AF3 is really opening the door to more targeted and effective drug therapies.
Okay, I have to ask about another example from the paper. It's a bit of a mouthful. A multivalent human antibody centaurin fusion protein.
Oh, yeah, that one.
What is that?
It's a really clever way to fight off infections, especially those caused by bacteria that are becoming resistant to our usual antibiotics.
So it's like a new weapon in our arsenal against superbugs.
Exactly. So this fusion protein combines the power of antibodies, our natural defenders, with these other proteins called centurins. They're kind of like tiny grappling hooks, and they can bind really tightly to specific targets.
So you're taking the best of both worlds.
Exactly. And the multivalent part means that this fusion protein has lots of binding sites, so it can attach to lots of targets on a bacteria at the same time.
Like Spider-Man, ensnaring its prey with multiple webs.
I love that analogy. And AF3 played a big role here, too. The researchers used it to model the structure of the fusion protein and predict how it would interact with its target on Staphylococcus aureus.
Staph aureus. Nasty.
Oh, it is. Can cause all sorts of infections. But AF3 helped make sure that this molecular Spider-Man was perfectly designed to capture its target.
That's so impressive. It seems like AF3 is helping us develop new ways to combat infectious diseases, and that's becoming increasingly important with antibiotic resistance on the rise.
Absolutely. It's a really big deal.
Okay, so I'm seeing a theme here. Whether it's understanding those basic biological processes or designing targeted drugs or developing new weapons against those nasty pathogens, AF3 seems to be at the forefront.
It really is. You see, AF3 is fundamentally changing how we do biological research. It's like having this incredibly powerful lens, letting us view the molecular world in ways we never could before.
And that means?
That means we can make discoveries and develop those life-saving therapies at a pace we've never seen before.
It's amazing to think about all the possibilities. But we do need to remember that AF3 is still a tool, right? And it has its limitations.
Of course, yes.
We've already discussed some of those limitations, but there's one more I want to touch on. AF3 predicts just a single static structure for a molecule. It's not like that in reality, right?
Right, it's a snapshot, a frozen picture.
Yeah.
But in the real world, those biomolecules, they're always moving, they're dynamic, they're
Yeah.
constantly changing their shapes.
It's like the difference between a photograph and a video, right?
Exactly. The photo gives you the structure, but you miss all the action, the movement.
And that dynamic behavior is often super important for how a molecule actually functions.
Right. So while AF3 gives us this incredibly valuable structural framework, it's important to remember it's only one piece of the puzzle.
Okay, so we've explored AF3's capabilities, its limitations, and some really cool applications. But what does it all mean? Why should our listeners care about this revolutionary technology? That's what we'll be diving into in the final part of our deep dive.
I'm looking forward to it.
All right, so we're back. We've spent some time with AlphaFold 3, exploring its mind-blowing capabilities, those quirks, those limitations. But now the big question, right? Why should you, the listener, care? What does it all mean?
That's what it comes down to, right? It's easy to get lost in all the tech stuff, but the real impact of AF3 is about what it allows us to do, understand and shape the world around us.
Let's talk impact then. Big picture.
Mm-hmm.
What are the implications? I'm ready to have my mind blown again.
Well, imagine this. A world where scientific discoveries happen super fast. AF3 is like this accelerator.
Like a turbocharger for research.
Right, exactly. Scientists can get super accurate models of these proteins and complexes so much faster than before.
Instead of months or even years of lab work.
Right, we're talking hours or days now.
That's insane. And that speed means faster progress in fields like drug discovery.
Exactly. And disease research. Think about it. We understand the structure of a disease-causing protein in a matter of days. Maybe we can start designing drugs to fight it that much faster.
So, new treatments. Cures, maybe, for some of the world's toughest diseases.
That's the goal. And it's not just about treating the diseases we know about.
Oh.
AF3 could help us uncover completely new targets for therapies, leading to breakthroughs we can't even imagine right now.
Okay. Now that's pretty amazing, but the implications go way beyond medicine, don't they?
Oh, absolutely. This tech could revolutionize all kinds of fields. Agriculture, environmental science, bioengineering, material science, even.
Let's hear some examples. How could AF3 impact agriculture? Especially with the challenges we're facing, feeding everyone, climate change.
Well, think about it. What if we could design crops that are resistant to, pests, diseases, even drought? AF3 could help us figure out how those traits work at the molecular level and then...
engineer better crops.
Right. Hardier, more productive.
That would be huge for food security. Global. No question. What about environmental science? Enzymes.
Yeah.
There's so much buzz about using them. To clean up pollution. Produce biofuels.
Right. AF3 could be really useful there if we understand those enzymes, the ones that can break down pollutants. We might be able to engineer them.
To make them even better.
Yeah, more efficient, maybe target specific contaminants.
So AF3, a tool for cleaning up the messes we've made.
Exactly.
Okay. Bioengineering. Designing new proteins.
AF3 is already having an impact there.
Really?
Researchers are using it to design proteins with very specific properties, like biosensors that can detect certain molecules or catalysts for industrial processes.
So it's like we're entering this age of protein design. Creating these molecular machines that can do pretty much anything we want.
Pretty much.
Okay, let me put on my speculation hat for a second here. The paper talks about applications in biology and medicine. But could AF3 be used in fields we haven't even thought of yet?
Definitely possible. Predicting those complex molecular structures, it's going to have ripple effects everywhere, not just life sciences. Material science, maybe? Imagine designing new materials with specific properties just by tweaking their molecular structure. Stronger, more flexible, conductive, you name it. It's like a molecular toolbox for creating new materials with tailor-made properties.
That's insane. New materials for construction, aerospace, electronics.
Energy production, who knows, maybe even for space exploration or building habitats on other planets. The possibilities are endless, really.
Okay, I'm officially mind blown. AF3 isn't just a tool then, it's a launch pad for a whole new wave of scientific discovery and technological innovation.
I like that, a launch pad. It really is a testament to human ingenuity and the potential of AI to transform our world.
Well, that brings us to the end of our deep dive on AlphaFold 3. It's been an incredible journey exploring this tech that's changing science, and so much more. We saw how AF3 is speeding up research, opening doors to new discoveries, and inspiring us to imagine a future full of possibilities.
It's been a pleasure to be here and to explore this incredible topic.
As always, we encourage you to check out the source material. There's so much more to discover, so keep those minds curious and stay tuned for more deep dives. Until next time, farewell.
Farewell.